Professor Chris G. Carter. Professor Carter is the head of the Fisheries and Aquaculture Center at the Institute for Marine and Antarctic Studies, University of Tasmania. Professor Carter's research focuses on nutritional physiology with the aim of understanding how aquatic animals use and waste nutrients. This encompasses understanding of climate change effects, growth under suboptimum conditions, and developing new ingredients and aquafeeds.
Thank you very much. I'm very honoured to be here. I do sincerely thank the organisers and the committee there. So I've been asked to give a talk about nutritional physiology, and I'm going to try and focus on the significance of nutritional physiology in commercial aquaculture. So this is going to be quite a rapid talk and go through a few components of nutritional physiology and just try and make some overview comments. So in terms of the outline, I'll just talk a little bit about what nutritional physiology is, comment on some perspectives and challenges, and then go through and end with some case studies to try and emphasise some points about why is nutritional physiology of relevance to commercial aquaculture. So this is a very interesting topic, nutritional physiology. I call myself a nutritional physiologist, and I thought I'd better have a look in the literature, and in all of the scientific literature since 1945, only 149 papers have actually used the word nutritional physiology in the title. So that immediately intrigued me. But on the other hand, I feel most people in aquaculture who are interested in aquaculture nutrition actually address a kind of nutritional physiological approach. They tend not to do straight nutrition, but have an approach that is nutritional physiology. So I've put some words up in there, which in a kind of very crude sense chart the history of nutritional physiology, which really started in the 17th century. So, this idea about combustion and use of oxygen, the fire of life, a famous book from the 1960s by Kleiber, calorimetry, indirect calorimetry, and an idea about the use of energy. So really, what we're talking about is energy use and the trade-offs that animals make in their energy use. And we saw a really nice presentation about osmoregulation and the trade-offs that the animal system was having to apply there. But thinking about the future, those more traditional views about nutrition and physiology are now kind of merging into the 21st century. So this is where we start to talk about all of those omics that we've heard today, starting with transcriptomics, but also nutrigenomics is a topic. And then trying to integrate that. So the key things there are, thinking about whole animal systems, thinking about the interactions between all of these systems and small, what physiologists do, what people who study aquatic animal health do. And what I think is a really key component is this idea about looking at our organisms, the aquaculture organisms, in terms of their interactions with the environment. And this is where it changes from nutrition. Nutrition is about the kind of pure, what's a requirement. Nutritional physiology is about what's that nutrient requirement in relation to the environment that the fish might be in. And they're not always in the ideal environment. So there's lots of little pictures down there which kind of reflect some aspects of nutritional physiology. Some of my favourite books I pulled out of my bookcase and just took a photograph, but you'll see some really famous names represented there. And Fish Physiology, a famous volume, number 7, has got everything you want to know, or what you wanted to know in the 1979. Okay, so as I've kind of said, it overlaps physiology, nutrition, health, genetics. It integrates biological systems and environmental factors. It really works best at a whole animal level. I think it's a very important way of thinking about things. It provides ways to understand underlying mechanisms. It evolves. This is really important. It's not an 18th century approach. It's a 21st century approach. It evolves and it incorporates new methods and approaches into itself. It provides a platform for combining, traditional stuff, the bioenergetics, simple measurements of oxygen consumption, for example, with the more advanced approaches, as we're kind of terming them nowadays, omic approaches. And I think, you know, relating to some of the talks we heard earlier today, it can inform selective breeding on many different levels. And I don't think selective breeding programs have really explored the levels that can be used. You can see there underneath just a kind of pictorial linking, you know, the animal in the centre in terms of growth, composition, things that you do in aquaculture, diet, health, age, and then around that other environment. But then perhaps just to formalise it a bit more in terms of this fora, we've kind of got this, you know, what is a way of thinking about this in terms of sustainable aquaculture? So I think one thing I wanted to emphasise was the production of scientific data. So scientific data to understand the influence of biotic and abiotic factors on production. So we saw some marvellous presentations yesterday in the Farmers Collective. This was a kind of a collaborative approach between farmers and researchers. The way forward with that is to kind of use the innovation, the imagination of farmers, but make sure it's done in a scientific context. We heard earlier today this importance about actually forming hypotheses and doing things in a scientific way. It's much better to know one thing correctly, i.e. get scientific proof, than perhaps know 10 different things, sort of, but not quite, and they may not apply. So I think part of this is actually re-emphasising the importance of doing science. I think one of the powers of, you know, looking at this, really looking at whole animals, is trying to identify solutions. And what are those solutions really about in terms of aquaculture? Well, I think at the core of it, it's about growth and growth efficiency. And in a way, you know, what is a healthy animal? It's all about a healthy animal is an animal that will grow and grow efficiently. And you can see down the bottom, I've just put a little pictorial that links the kind of more nutrition aspects. So commercial aquafeeds is going to be a way that we actually manage this growth and growth efficiency. And that's where a lot of nutrition research gets done. Towards the commercial aquaculture side, there's going to be a tendency to focus on more the kind of physiological work. And then those two, in a sense, overlap. And that will inform the process. This is just about, you know, you can see the arrow aquafeed going from the producer to the commercial aquaculture. It doesn't say anything about all the interchange. So there could be lots of arrows there about how knowledge is created and how people do research. Okay, so nutritional physiology. I just wanted to make this comment because I think one of the most important things that we're considering now is not only the environment and how that interacts with our aquaculture organisms, but it's also about the change, climate change, environmental change. And the key concept here is that we're having to look at, or certainly in many cases, we're having to look at our animals in suboptimum conditions. So in my field of nutrition, for many years, people had small animals in glass aquaria in perfect conditions and said the nutritional requirement is this. But what we're now having to do is redefine what those nutrient requirements are under suboptimum conditions. And I think that's a really important, you know, task for nutritional physiology. You can see this idea about temperature. Malcolm Jobling, one of the really great nutritional physiologists, put it together in terms of that blue figure. Most of you will have seen that, but I think it's a really nice way of just pulling together this idea about temperature, how it drives feed intake, how that is an advance in a sense of growth. There's an idea of an optimum, it peaks and then it falls off there. But a key thing for me is the efficiency, the difference between the feed intake and the growth. And that's one of the kind of biological features, but also a characteristic for aquaculture that's really key, I think. So down the bottom there, you know, commercial significance and solutions around climate change. You know, different species. We've talked about aquaculture looking at many hundreds of species. Part of that is managing changes in the environment. A lot of talk nowadays about, relocating farming, the use in selective breeding, and then more things to do with actually refining practice, and then the nitty-gritty of kind of nutrition, changing some of the formulations, etc. Commercial significance and scale. I thought it was worth putting this up. This is a reflection of the research that we've been doing on rock lobster aquaculture. So we feel it's been a really good achievement of ours that we closed the life cycle of rock lobster. But I just wanted to pull out of that something to do with scale. So first thing to do with scale was the size of the technology. If you're going to achieve something that's meaningful to aquaculture, then it would need to be on a scale that relates to aquaculture. This is what was nice about yesterday. You saw people working on farms, not in test tubes or small aquaria. Time. You actually need time to achieve these. And so down here you can see we kind of started in 1998 and it took about 20 years to achieve this. A previous slide today showed what's happening with northern bluefin tuna. It took even longer, more than 30 years to close the life cycle for that species. And then the third one is scale in terms of investment. If you're actually going to do or answer some of these big questions, you need millions of dollars to actually achieve these. So scale is an important thing in terms of commercial significance and the way you link it with the tools that you use. So I've just listed some of those in terms of how important benchmarking is, the use of model species, how can you transfer information. A good example is Atlantic salmon. There was a massive Atlantic salmon industry in the world, and that was before we knew the amino acid requirements for Atlantic salmon. So we were able to produce tons of Atlantic salmon with no knowledge about the requirements or guesses. And then this idea about steps to commercialization. The thing I wanted to pull out there was an idea that in any process, you must have a kind of pre-commercial validation. You can do the experimental stuff, but you must have on-farm stuff, but not as a production, as an actual test of your concepts. Okay, so I've kind of covered a bit of the context just to try and place this in a sort of flavour of where we are in this conference. Now I thought I'd just work through some case studies. I've tried to find case studies which are relevant to brackish water or at least tropical aquaculture. A lot of work I do is on Atlantic salmon. I don't think you'll be growing Atlantic salmon in India very soon, unless of course you move offshore into cold waters. So hence I'm going to look at Asian sea bass first. So I really like this figure because it says a lot about a euryhaline species, barramundi, and again just referring to the earlier talk about osmoregulation. Euryhaline fish are going to be quite special in their ability to cope with varying environments. And the key figures I'd like you to look at there are the two on the right. So I've termed them PPV, which stands for productive protein value, but what it really is an expression of how efficiently nitrogen's retained, and the bottom one is how efficiently energy is retained. But the key thing for me there with this species is the plateau of that hill and how it extends over a very wide temperature range. So barramundi, Asian sea bass, are maximally efficient over a very wide temperature range from nearly 25 degrees right up to maybe 35 or 36 degrees. And I think that kind of defending efficiency would be a very important evolutionary strategy for those fish. It's a way that they actually make the most of the food that they get. I also think it's a really important component of how you would judge aquaculture. And what we should be doing is looking to maximize species efficiencies. So obviously we maintain a high growth rate, but we also need to be looking at fish being highly efficient. So if you look where that line peaks, that's actually saying that 50% of the nitrogen and nearly 50% of the energy is retained by those fish. And as a previous speaker said, one of the marvelous things about cold-blooded ectothermic animals that we grow in aquaculture is how efficient they are at converting nutrients into growth. So why are animals so efficient? What are the underlying mechanisms? Again, remembering this is all about a trade-off with energy. One of the things I've been interested in is actually the utilization of proteins and protein turnover. So you can measure synthesis in various ways. Measuring degradation is a bit trickier. You can use labels for synthesis, but there's different degradation pathways. But some people have said, degradation costs about the same amount of energy as synthesis. So So that's the first point. The second point is that it is really energy expensive. So if animals get it wrong or they use protein synthesis for other things, like managing an influx of amino acids, then they're not going to use that energy for growth. So that's a kind of driver of this. And you can also mix it with other, metabolic processes. So to me, this is kind of at the heart of nutritional physiology in the kind of bioenergetic sense. And why it's useful to aquaculture is that it kind of underpins the provision, the use and provision of energy, amino acids, proteins, etc. And that, of course, underpins how the fish grow and how efficient they are at growing. And I really believe that those are components that need to be incorporated into selective breeding programs. But we'll be able to understand the mechanisms and understand why those are such good processes to think about. So this is a case study, if you like. What happened to barramundi when they were exposed to very high temperature, an elevated temperature where they start behaving suboptimally at an animal system sense? First thing that happens, if you look feed intake, FI, that goes down to about 45%. You get a reduction in growth efficiency by about 60%, 0.6, so change in efficiency from 50% to less than 30%. And some of the reasons are to do with, down the bottom of that, you see protein synthesis, PS, that goes down by 50%. So the animal's actually responding to an environmental challenge by down-regulating an energy-expensive process. And then here the hypothesis is that protein degradation goes up. And that was actually captured by gene expression stuff, looking at one of the cathepsins. Cathepsin D expression went up. So that's actually telling us that the animals have switched on a specific degradation pathway. And so the hypothesis about this was that they are using their protein, as was actually hypothesized earlier, using their protein as an energy source. They have no alternative. So commercial outcome, perhaps it leads to improved feed formulation in relation to suboptimum conditions. It confirms barramundi, I believe, a really useful model. So we kind of we're looking at salmon, barramundi, trying to use those as a kind of model systems. And if you have a model species, it's really useful because it allows you to go through more rapidly some of the fundamental research to kind of kickstart and provide information for your particular aquaculture endeavour. So that was kind of an example there. Asian sea bass, a stressful environmental temperature, and just some way of using nutritional physiology to explore that. Next one I thought I'd put up was something about rock lobsters. So we've been working on this. We've reduced the life cycle. We can produce the tropical rock lobster here as shown by the life cycle. But the particular problem I'm interested in is of course developing feeds. So we've kind of done the larval feed bit. We're now focused on the juvenile feed bit. So you can see at the moment, and most of you will be aware, that rock lobster aquaculture in the world is basically used as so-called trash fish in fairly standard ways. And I've seen, the people here are working on formulated feeds for rock lobster. But what kind of work could we do adopting a nutritional physiology approach? So I'm still kind of thinking about those cycles of metabolism over 24 hours. The figures are really just showing you oxygen consumption. They're showing you carbon dioxide excretion. They're showing you nitrogen excretion. And it's going back to, a relatively traditional bioenergetic approach. But if you combine knowledge about nitrogen, carbon dioxide, oxygen consumption, then you can start to actually think about substrate use over 24-hour cycles. So the figure on the left is actually a representation of the change in substrate use over 24 hours. So it's kind of showing you a shift between protein, lipid, and carbohydrate. The red is the carbohydrate. About 4 to 12 hours after feeding, there seems to be an elevation in carbohydrate utilization. So very interesting, very nice for me as a physiologist, a nutritionist, to play around with the lobster. You can see it in the respirometer at the top. But the kind of commercial outcome is by understanding at a kind of fairly detailed level how this organism is using the feed that we have in terms of which substrates, then that will more rapidly progress the development of feeds. We've got a tool that you can use in a short-term way to understand how feeds are used. So we can manipulate feeds and we can have a look at the effects of those on metabolism, as opposed to the more standard approach, which takes much longer, of actually growing the fish or lobsters for a long period of time. Okay, moving to a different scenario, thinking about nutritional physiology. The kind of theme here, though, is energy expenditure under suboptimum conditions. What you see there is a representation of using sensors, both around a salmon cage, within a salmon cage, and also on the salmon themselves to understand their behavior, their response to hypoxia, to elevated temperature, things like that. So it's really just, and we heard the use of the term sentinel, we had sentinel fish. It's really just about understanding the kind of feed energy expenditure dynamics of fish in a real situation. Again, commercial outcome, combine real-time data and integrate that with salmon models, and that will feed back into tools to manage farms, make decisions about how much food based on kind of real-time assessment of what's happening in a cage whilst you're looking at it. But for me, it comes down to understanding the stresses and the energy expenditure and the way to supply energy. And coming to the end very quickly, this one is about digestibility. So lots of people measure digestibility in not very good ways scientifically. This is about doing it on a commercial scale. This is about taking a big scoop net of salmon out of a commercial cage, stripping the salmon in a large boat by those cages, and getting accurate measurements of digestibility in situ. But the other key part of this is using commercial feeds without changing them. So you have to find an internal marker to do that. So 2 points there. Actually measuring digestibility properly, finding an internal marker that's in commercial feeds so you don't disrupt the feeds themselves. And then you have a tool which gets you to some important commercial outcomes. So again, it will help improve feeds. It will help improve those feeds for the environments that the fish are in. But probably more importantly for the industry, it will help with environmental models. And the important thing for industry, it will actually allow regulators to set targets much more accurately if you have much more accurate data. So at the moment, in Tasmania, for example, we just use one figure for digestibility. It doesn't matter what the temperature or the size of the fish. With this kind of method, we can actually go out and make much better models. So really important commercial outcomes there. So I'll move to the end. So very briefly, as I've tried to say, it's probably what most of us do as biologists when we think about feeds and feeding, a kind of adopt a nutritional physiology approach. It really means that we're thinking about animal systems. We've got lots of different techniques, there's lots of layers. I've tried to put a table down there and put my examples into that table in terms of the parameters that we consider, practical approaches, and then some of those commercial outcomes that I've told you about. So that just provides you with a summary. And then the conclusion is really that nutritional physiology is essential in the toolbox for advancing sustainable aquaculture. I did write this initially as some scientists need to remember that aquaculture concerns whole living organisms, but I thought I'd just be a bit more polite and just say aquaculture concerns whole living organisms. But I feel we forget that sometimes. We get a bit carried away with the technology and the new technology and forget some of the basics. But as I said, I was pleasantly reminded of just how people do consider organism biology in terms of their farming, having seen all those marvellous presentations yesterday. So thank you.